Yeah, no, very interesting. And do you see any similarities other than what we discussed already with the astrophysics and the EEG analysis? How do you usually analyze the data? Do you see some different patterns than others? Do you approach analyzing the EEG somehow differently?
With the EEG, when mathematically, when you first start to tackle it, it's Fourier analysis, and Fourier analysis is everywhere across the sciences. So you can use it to pick out particular frequencies, and in this way, it's similar to looking at music. I also have a strong interest in music. I'm a cello student and I love going to the symphony. And so I think a lot about music, and I love pulling music into MATLAB and just playing with it. And so when you first start doing the analysis, it's a lot like that. It's using Fourier analysis to break out different frequencies. But one thing that's different about EEG and the brain and working with neurons is that it's easy to think, it's easy to look at the EEG data. If you look at it visually, it looks like music. It looks like that type of signal, like a vibration. It's easy. So, physicists, we all start physics thinking about a vibration on a string. So, it's easy
Mm,
to look at the EEG and think this is a complex vibration on a string, and it is not. So, the thing with a neuron is governed by an action potential. So basically, it's a chemical reaction. If it builds up enough, that the neuron fires off an action potential. So you can think of a neuron as being an on-off switch. It's a blinking light. It's capable
mm,
of sending out exactly one signal, that action
mm,
potential.
mm,
So it is not a vibration on a string. So what you're looking at when you're looking at an EEG is what a neuron can do when it's talking is okay, it can send out, it is a beeper capable of producing one note. But the way that it communicates different messages is the frequency of that beep.
mm,
So a quiet neuron might be going beep, 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 while an excited neuron is beep, 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 beep. So what that looks like when you're looking at the EEG is you're seeing an entire population of neurons. And when you see higher amplitudes in the EEG, it's not like a vibration on a string where you're seeing a higher vibrational mode in the string. What you're seeing is more neurons going beep, 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 and they can produce exactly one beep. And I try and keep that visual image in my head when I'm processing this data that it's not like music where you've got different notes. You've got one note and what you're looking at is the frequency of neurons blinking. It's like a crazy Christmas tree. So that's sort of from a physics standpoint, like that's how I image the data, imagine the data in my head when I'm working with it. And then the question becomes, well, what can we learn from this? And when we're specifically looking at the slow oscillations, you're seeing entire populations of neurons in the EEG that are having action potentials at the same very slow, very relaxed rate. They're doing it together. What does that mean? And that's what we're
mm,
trying to understand.
mm. No, I think I will put the crazy Christmas tree in the episode title. I think that's that is clever. I think that's a good point. I think we often forget to visualize the signal like what it is. We see just a lot of curves in different channels, and I think the visualization is the key. Do you try to visualize or have this kind of some kind of analogus for a Christmas tree or something with also other signals, for example, EMG? How do you visualize? How do you understand EMG?
EMG, I don't have probably the same strong visual image with that because the EMG with that I'm pretty much just looking, is there activity? With the
Mm,
EMG, I'm more concerned, especially since we're working with data from the animal lab, I'm more concerned about looking for artifacts and looking for noise.
mm,
So there I'm more concerned with differentiating good data from bad data. And
Mm,
with that, because I have a very specific question I'm trying to answer with the EMG. With the EEG, there's a lot more exploration going on because we're
mm,
really trying to understand what is happening inside the brain. The EMG, because we're not studying muscles, the only reason we have that there is to try and see if we're seeing the signs of REM behavior disorder.
mm,
Are the muscles twitching when they shouldn't be twitching? So
mm,
in that case, my from my data analysis standpoint, my first task is to look for bad data,
mm,
make sure that what I'm seeing is real, and second, just seeing if it meets a certain threshold. Is this a muscle twitching when it shouldn't be twitching? And then making sure that I line that data up correctly with the EEG that I'm using to see if this animal is in REM sleep,
mm,
making sure that lineup is correctly. So that's really all I'm doing with the EMG. So I'm not investing
mm.
a deep visualization there because
Yeah,
I'm really just looking for, is it on or off?
yeah. So basically, you are just from EMG kind of looking, is the brain sending signal to the muscle that causes the
Exactly.
activation? Yeah, that's very very interesting. And maybe continuing with this theme, you said that you are very interested in music and you want to look at the signal of the music, which I'm also doing when editing the podcast. So I have used to looking the sound, and it's quite interesting that, for example, many people say like before they start,
Right. Yes.
and when you're editing the podcast, you start to see that this shape is always the arm, and then some people say and they swallow and you see, and then you can delete without even listening because you know that
Right.
that's the sounds that you can delete. So about the music, how does it help in science to be excited about music and understanding the sounds that are in the music?
So I've pulled music into my work recently. So I also teach an undergraduate physics class this term, and it's a non-major class, so I'm trying to make it very application heavy. Like, this is how we apply physics in our lives. And for one of my classes, I brought in my cello and a microphone and played a few notes, and we pulled it into MATLAB to look at it. And so from a physics standpoint, what I was demonstrating was the cello is a vibrating string. This is what a vibrating string looks like. Here are the different
Mm,
frequencies. And we did the math on the board, how to calculate the frequency of a vibrating string, that sort of thing. But from a neuroscience perspective, and I pointed this out in class, so I played the string on the cello, and then I also played using MATLAB. You can generate pure tones. So I generated a pure tone that was the same frequency as a string. So the cello, generally people find the sound pleasing, and the
mm,
pure tone is annoying. There's a reason why we make alarms and warning buzzers that electronic, single
mm,
tone, because it's really annoying and you want to make it stop. So we used Fourier analysis to compare the two. Like, why is this electric tone annoying and the cello sounds rich and beautiful? Well, if you do the Fourier analysis, electric tone has a single frequency. It's a
mm,
spike. You get this one
mm,
frequency. The musical instrument, like the cello, if you do the Fourier analysis there, you get all the harmonics because you get the main vibration, the entire string. That's the main frequency. But you get all these harmonics because basically every possible vibration that string can produce also appears in the Fourier analysis, and you end up with a much more complex sound. And in that way, it's a lot like the human voice. Like, the human voice is not a single frequency, it's many frequencies layered on top of each other, and our brains like to listen to voices. So that was visually just a way to use physics to say, hey, why is this sound pleasing to us and this sound really annoying? And another topic that I've been interested in and just reading about, so our group at Sharp at OHSU, we're involved in a lot of different neuroscience studies, all with the focus of improving lives for people. And one of the conditions we do think about is Parkinson's disease. And so, reading about Parkinson's disease, a lot of people, and this is true with persons with Alzheimer's disease as well, they just sort of perform better when they're listening to music. It's easier for them to walk. It's easier for them to dance. It's easier for them to exercise. People with memory, people who might be struggling to find words, can sing songs. So, what is happening with music? And there are studies happening where people are trying to figure out, like, what is the brain doing when it's listening to music that's different, and how can we use that to help people? So that's we don't have an active study on that right now, but I just think it's an interesting topic of research that the brain does something really unique when it's listening to music, and we don't quite entirely understand what that is.
Yeah, that's very interesting. And I think when they hear rhythm or music, a person with the Parkinson's, they are walking better. But it's also rhythm, left, right, left, right, left, right. Like walking is a rhythm, and you need to activate this. And I had a podcast recording where we discussed also that there are three animals that can dance, and one is human, and one is Asian elephant, and one is a parrot, and they are the same animals that can talk. So they can talk. Asian elephant, I think they show that scientifically that they can say five. You can teach five words in Korean to them. But anyway, I think the point was that somehow our hearing and our motor cortex was connected, because we need to change our movements in our throat, that we can actually make the sounds that we are able. So there's a connection between the ear and the motor cortex that is directing, and that's how somehow when we dance we are using this same connection, that the music that's coming to our ear we use to move our body in the same rhythm, or something like this. But I think it's very, very, very fascinating, this link between music and motor function. And yeah, also when you said this single tone, and like if I give examples of podcast editing, sometimes there's from a connection there's some noise coming, and it's really annoying, and then I try to edit it out. I'm doing the fast Fourier transformation, and I see the signal, and I try to find that what is the noise that can I just cut it. And it's super difficult because human voice has almost
Yes.
all the frequencies.
Yes, definitely.
Yeah. So let me see. We have discussed a lot of different things. I have my notes, 3 pages already. Maybe we discuss about this, that you have the human and animal studies at the same time, and you can look how do you use it. What do you see as the biggest advantages of this setting?
So with the animal studies, we can observe, we can have more control over our animal studies. And I'll explain what I mean there by talking a bit about the human studies.
Mm,
So my first experience when I was a postdoc with Dr. K, he runs a project called Orcatech. And it is this project of observing people, older people in their home environments, going about their regular lives over long periods of time. There are people who've been volunteers in this project for more than 10 years. And the way that they're observed is these very simple little motion sensors are placed throughout their homes, and you can just pick up that where people are moving around. It's very, very simple data. It's a simple timestamp. Motion was detected.
mm,
So, as a data scientist, my role was to take this very simple data and turn it into observations about the person's circadian rhythms and their life patterns and their activity and that sort of thing. And so, one of the things I learned, so you had, in order to do this, you had to make assumptions. So because this data, again, very simple, just a timestamp. Oh, the motion detector blinked at this exact moment. So you would have to make assumptions like the person, if they go into their bedroom and they're in bed and they're asleep, and we can estimate their sleep time. The thing that I learned from working with humans is that any assumption you make is going to be wrong. People will have all kinds of unusual habits that you will never be able to predict. Very quickly, I learned a lot of people sleep on their couches, a lot of people sleep in their recliner, or a lot of people get up and eat in the middle of the night, that sort of thing. So you would have to build those assumptions in, and they would automatically be wrong.
Mm.
So people are really difficult to predict. So from a data science standpoint, it makes it really challenging, interesting to work with, but challenging. So in the animal lab, if we're doing studies of sleep and circadian rhythms and activity, we have an environment we can absolutely control. We know when the lights are on and off. We know what the temperature is. We know where those animals are. And so that makes it really useful to study circadian rhythms and how that affects health and sleep patterns and things like REM behavior disorder, because you know exactly what that animal's
Mm.
daily activity has been. And we can do things. We also do studies looking at social behavior between animals. One of the animals we work with is a prairie vole. And the reason why prairie voles are interesting is they're unusual as rodents because they bond in a very human-like way. So they're a good way to study how bonding affects development, and the relationship between social interactions and sleep. Like we're finding that we know that, we all know that sleep deprivation is terrible for humans. Everybody's experienced it. But in these animals, these prairie voles, we can see exactly how, especially early in life, how does sleep disruption and sleep deprivation affect how they behave as adults towards each other. And we do see that it has an effect. So the way that helps us is if we're looking at REM behavior disorder in mice, which is something we are looking at. Well, those mice, I know exactly what their sleep patterns are. I know exactly what they've been doing every day. I can measure over days, their EEG patterns, that sort of thing, because they are a captive audience that I can see what they're doing, which is much more difficult in a human. So we also have these studies with our human volunteers, and the human volunteers will come in for a night of polysomnography. They will come in for MRI scans. But in that case, we're only capturing a moment in their life. We're capturing one night. And so, but by combining the two, we can use the animal data to learn about the human data. And so they complement each other really well.
Yeah, interesting. And you've been doing quite a bit of studies of circadian rhythm, and you said that it's the assumptions what you make of humans is usually incorrect. How do you see the technology of measuring different variables that relate to circadian rhythm? What would you like to measure that would really help the understanding of circadian rhythm in humans? What would we need to measure better that we would understand them better?
I think one thing that's really challenging is studying when people are actually asleep. So one of the calculations that I've done, I took a look at the data from the Orchitect study over these long periods. And so these are older adults, and things happen with circadian rhythms as you age, even during healthy aging, but especially if you develop something like Alzheimer's disease, is your circadian rhythms start to weaken. What happens is you have naps during the day, you have wakenings during the night, you have a lot more sleep fragmentation. And so one of the things that I found looking at this data is a lot of times in circadian rhythm studies, people try and take this sleep active data and fit it to a cosine curve. So during the day, and then we're asleep during the night. We're awake during the day, and we have this perfect, beautiful 24-hour rhythm. It's really not true for anybody. And If you have disrupted circadian rhythms, it becomes even more difficult. So, one measure that I liked to use was interdaily stability, intradaily variability. I found this a very useful measure, and these were metrics that were designed to look at circadian rhythms. So, the interdaily stability, instead of trying to take your data and force it to be a cosine, what it does is it just, you take your data for a single person and you break it into 24-hour chunks, and you look at how much do each of these 24-hour chunks look like each other? How
Mm.
similar are your days? Do you have a regular pattern? And so that's one measure we could use for circadian rhythms. The other is intradaily variability. Intradaily variability is looking at your fragmentation. How often are you transitioning between being awake and asleep? And are you waking up a lot at night? Are you taking a lot of naps? We have this ideal vision in our heads. Everybody thinks, oh, I'm going to lay down, I'm going to sleep for a solid 8 hours, and then I'm going to be awake the rest of the day. And that happens for nobody. Everybody has awakenings during the night. But especially as you age, and we see this in people with Alzheimer's disease, they have more sleep fragmentation, more
Mm.
transitions between being awake and being asleep. And I found this was really interesting. So with the Architect data set, again, we had years worth of data. So you could really see, I found some cases where just by looking at this data that was entirely from these little motion sensors, I could see changes in people's lives. I saw one case where there was a woman and you could see that her patterns had changed. And I had access to her health, her personal, the participants once a week would fill out a report talking about their health and other events in their life. And it turns out she had been diagnosed with a medical condition, and eventually the treatment worked and she improved. And if I looked at her data, I could see the same pattern. She had increased fragmentation, her days were not as predictable, and then she recovered, and it came back. Another really interesting case, and none of this is published, it was just thing anecdotal, but there was one gentleman looking at his data, and his intradaily variability decreased. He had fewer of these transitions. His sleep was becoming more stable with time. And I was like, wow, that's really cool. So I looked at his reports, and he had written a report saying that he had started using lavender essential oils at night. And in his report, he says, I feel like my sleep has improved. Like I'm sleeping better. I was like, the data is, yes, you're correct. Your sleep has improved. And I was curious enough about it that I did a literature search. And there is some literature, there are some publications supporting the idea that, especially as you age, lavender essential oils can help you sleep. Again, it's very, very preliminary, sort of anecdotal data, but it was just interesting and fun. So, in terms of measuring people's sleep for long term studying people's life patterns, we have to accept the limitations of what we can do. In
Mm.
order to really, right now, the gold standard is polysomnography, which is you have an EEG, you're measuring people's respiratory rate. You've got it's pretty uncomfortable, but it's the one method that really reliably allows us to tell, well, what stage of sleep are you in? There's no
Mm.
way we can do that for people over a long period. But I think if we can get technology wise, like if we can get better at differentiating between sleep and rest, in a lot of the publications I've written, I've had to make the caveat that with these technologies, these unobtrusive technologies, I can't 100% tell that somebody is asleep. So we'll say this is resting behavior. This
Mm.
person is still, they're resting, but I can't tell you 100% that they're actually physically asleep. So
Mm.
I think that that's one challenge, is if we can get better at telling when somebody is just laying still, maybe reading, watching TV in bed, or if they're actually physically asleep without being obtrusive. That,
Mm.
I think that's a really interesting problem.